it is my great pleasure to welcome and introduce to you Dr. Jie Mei, who is currently a project assistant professor at the International Research Center for Neurointelligence at the University of Tokyo. After completing her master's and doctoral thesis and studies in Paris École Normale Supérieure, sorry for my French, and Charité Universitätsmedizin in Berlin, Dr. Mei worked as a postdoctoral fellow at Western University and the University of Quebec in Canada, and her research focuses on learning and behavior in both health and disease, and she is using both experimental and computational techniques in her studies. Her interests include machine learning and deep learning for the analysis of neuroscience data, biologically detailed neuronal models, and neuro-inspired artificial intelligence. But for today's talk, the main topic will be introduction or bringing the conventional traditional tests closer to the animal home cage, and namely radial maze. And with this, I'm happy to give screen over to you, Jie. Thank you.
Thank you for introducing me, and thank you for having me. I hope this is going to be an interesting chance for all of us to understand how behaviorally relevant automated testing devices are being introduced into the experimental paradigms of, like, for example, different behavioral tasks. And I'm going to start sharing my screen shortly. As you have potentially read online already, today I'll give a talk on maze. So maze are really widely used in our experimental studies to investigate animal behavior, especially in spatial navigation. And traditionally, when we are doing maze experiments, we do a lot of constraints on food or water to motivate the animals to enter and to navigate within the maze. And today we wanted to highlight how we can potentially introduce automated, stress-free and relatively more stress-free testing devices to this paradigm to understand how spatial navigation and memory are playing a role in maze, for example, in animal cognition. So a bit about myself, I'm currently working in Tokyo, but I definitely have quite an international background. I completed, so this is a talk about a lot of the stuff I've done during my PhD in Berlin. And I think for most of us, we are sharing our passion and motivation for improving animal testing. But in case you have anything that you don't understand or any background information you require during my talk, just feel free to let me know anytime. I can take a pause to explain the things you want to know. And we can just dive right in, I guess. So as experimentalists, and assume most of us are, we have seen a variety of mazes that have been used to determine cognitive characteristics of animals, rodents, primarily, and there is a big variety of them. And for today, we are focusing on one of this, but I guess it's a nice overview for us to understand the variety and the need for automation for most of these apparatus. So we are focusing on radial arm maze for today, which is relevant to spatial memory testing. And for today's talk, we're focusing on two paradigms. The first one is working memory, and the second one is combined working reference memory. So for this task, when we are doing the manual version of it, we can see that we require a lot of manual habituation testing. For example, for the daily entry to the maze, we have to have an experimenter introducing animals to the maze, as well as prior to the experimentation, we have to have the same person there to habituate the animals. So there is definitely a need for a lot of interactions between animals and experimenters. In addition to that, we often have daily experiments for these protocols. So mostly we have 1 or 2 testing daily over a sequence of sometimes a week or sometimes more than that. And in addition to this interactions, we also introduce a lot of stress to the animals because we have to make sure that they're motivated enough to navigate within the maze. And for that, we have to food deprive more water, restrain them so that they have enough motivation to search for rewards or just for food when they're first introduced to the maze. So these together actually are truly introducing a lot of anxiety and potential stress to the animals through testing. And it's not only require a lot of intensive handling and efforts from the experimenters, but what's more important is that it can be a potential confounding factor and bias the results we are seeing in these tests, and can sometimes, for example, for food or water deprivation scenarios, can really compromise the brain biology or morphology of some animals, as have been suggested in some studies. And these are all like the confounding factors we wanna try to avoid, especially during spatial testing and cognitive behavior testing. So there's a need on the level of animal welfare, for example, and in addition to that, in pursuit of truth and we want to validate our scientific findings to make it reproducible. We want to also minimize these kind of handling stress, as well as the change in the animal's biology. So under this motivation, this is kind of interesting, as I only make a screenshot of the radial arm maze. We are trying to already, even before our studies, there are some people trying to introduce different factors or mechanical components or sensors to semi-automatize these mazes. For example, there are previous attempts to introduce pallet sensors or detectors or like different pressure sensors to track the animal's behavior automatically. So these are some studies even from the 90s. So we can see like we have a long line of automation of behavior testing standing in front of us. And in addition, some people also try to make the mechanical parts of the radial arm maze more controllable using a software, or some people have also tried to automatize data collection and analysis using recording, and they can pull out these recordings to really do a software-centered analysis. And for our study, I mean, like we tried. In addition to this, we tried something else. We want to not only to semi-automatize a maze, but fully automatize the testing, throughout the testing, not only the testing, but also habituation using our devices. So you'll find a description of all these, apparatus we use in these two studies on the lower right corner. And our motivation, in addition to that, is, to have a really well understanding of how these behavior testing play a role, as well as whether they're feasible to be introduced to our studies. So if we have something that's super fancy and looks okay, this is fully automated, but it's not like really allowing the animals to study about the spatial layout of the maze, or it's hard to contrast the results we have in our maze with the classic radial arm maze. So what's the point of that? So we also want to have a really clear comparison and validation of our studies in comparison with previous studies on classic radial arm maze that's mostly done manually. So this is our design of experiment, and we are kind of trying to understand these different aspects. So we are doing usability testing for different age group to see if this apparatus can be used for different animals of different age. So in both our studies, we are trying to do that. And then we wanted to understand if this testing can be done for animals that are suffering from, for example, systematic inflammation and other cognitive disorders. So we have done that in study 2. And also we wanted to see if we can use this automated testing for different paradigms for memory, exploration. In our case, it's going to be the working memory paradigm plus a combined working reference memory paradigm. And this is how we initially planned out. And this is, I think most of us, we don't need that, but actually you see like on the lower figures, there's a, explanation of how these paradigms are done individually. So for the working memory paradigm, we are baiting with all the arms with the reward, and for the reference working memory paradigm, we are only baiting the animal with some arms rewarded, and the rest of the, for example, like the attempt to enter other arms were a bit more invalid in a way. They're not returning the animals with rewards. So, and this is part of our apparatus. I mean, we are mainly discussing the automated radial arm maze, but for the habituation, I think it's going to be a bit of useless. We have, again, like a lot of human intervention during this habituation phase. So we are also trying to automatize the habituation. So how do we do that? So in contrast to the traditional manual based habituation phase, we are introducing such a device, and we are kind of customize it using thanks to our animal technicians, we are having these climbing pieces that's commonly seen, for example, in our studies using automated maze, and we are placing this kind of customized lid on top of classic animal housing home cage, and the animals can feel free to really ambulate within such a maze to understand, okay, what are the climbing pieces look like and how it feels like to acquire such rewards, mostly like sugar pellets on top of it. So they're more customized to this climbing activities and the layout of such, for example, climbing assistive tubes. They are I think they are a bit more assured that, okay, the environment within this novel device is safe and they can feel free to navigate the space and acquire the rewards. So we are introducing the sugar pellets from the top, the burr hole on the top on a daily basis. So they learn how these pellets are looking like, how they smell like, and they know like, oh, this is something good, I want to pursue that. So even after they are introduced to the automated maze. So this is the first part of the experiment. Very importantly for us is to have such an automated habituation without any intervention from the experimenters. I think this is quite important. And after this habituation phase, we are introducing again, finally, the automated 8-arm radar maze, and this is how it looks like. This is just a skeleton of it. Of course, by the end, you have a lid on the top, so in case the animal just try to escape. And we have 8 arms extending from the central platform, and each is a half a meter long. And by the end of each arm, we have a sugar pellet dispenser that's automatic. So it's pretty much like we are using RFID, radio frequency ID based detection technique. And whenever the animal enters and the ID is registered, is not foreign to this machine, it dispenses a pellet by the end of the arm, depending on if the arm is baited or not. So in case it's not baited during the reference memory testing, is no sugar pellet will be dispensed. And also we are, this is how it looks like. On the right, you see how we are connecting to the home housing cage of a bunch of animals to this device, and we will definitely have a closer look later. But this is how the animals are mostly introduced to this ID sorter, which allows animals to come into the maze based on their RFID again, and then they're climbing up until when they're reaching this central platform, as you can see on the right, and start exploring the whole maze. And then on the right, we have shown how this looks like when you have the lid on, and we put a bottle on that, so in case the animal just escape. And then it's these are some of other mechanical parts that's important to our experiment. So first, how are animals determining the direction? So we know the rodents are really sensitive to tactile cues, and so we are introducing such a panel, and on the different directions we have different sensory cues and tactile, of course, in this example. So they know like I'm in this direction, I'm in this direction. And so we use this as a potential reference for the animals, for especially in spatial learning. And the second part is automated palette dispenser, as I've introduced. So it's really designed smartly, and this is really sensitive at the beginning. So we took a lot of time to tune it to make sure it's properly working and dispensing one palette instead of random amount of pallets. But now it's all working nicely. So when the ID is showing up, and I think there's a thing to connecting the the stuff up there, and one pallet is released down to the end of the arm. So the animal has to really ambulate through the end of the arm to reach this pallet. And after that, you see like connecting the home cage and the the automated maze is ID sorter. So this is not only a way that we are monitoring the animal's activity. So again, this is fully automated. So we have to have a way to record the animal's activities, not only about their daily activity and exploration of the maze, but how which animal exactly this is activating, is having activities in the maze. So this is the thing. That's why we're having the ID sorter. So this register, we have a pre-registered RFID sheet. We know this spatial, and the machine knows this batch of animals are about to enter the maze. And when the animals show up, the door open, and the animals in these the center of the tube in between the two doors, and the ID is checked. And then when we know that this is animal we are training for this paradigm, the door on the right is open and allowing access to this to this maze. This is also a way to control the daily entries of the animals, because during the trial we had some animals that are super active and they want to enter this maze all the time, maybe because there's something nice and sweet waiting for them. So we want to control the entries of animals on a daily basis up to, for example, 10 or 5 attempts. So this is also a way to monitor and to control the daily activities of the animals. So we are training them in a more fair way, instead of just train one animal one time per day and the others like 200 times per day. So this is how it looks like. And I hope this, okay, it's working. So this is a demonstration of how the animals actually, so you see this is the home cage and the animal just jump onto the tube. And then, yeah, so this is the left door of the ID sorter. Then the ID of the animals is inspected in the middle of this. And in case the animal has not reached the daily quota of radial arm maze entries, so the door, it had to wait a bit. And also, like in case there are 2 animals coming in a sequence, we want to make sure there's only 1 animal that enters, and sometimes it happens, and we have to wait for a sufficient amount of time, to open the right door to allow entry of 1 single animal, not 2 of them. So this is also like a safety concern and for training purposes. So the animal is not so afraid or worried because the animal is really familiar with the whole environment. And you see like the upper climbing piece is really close to what we have in this cage. So the animal is not afraid at all, just goes up. And this is one of the demonstration we have regarding, how this can be done. There's one other I can share with you. Just a second. Okay, we have this online. Here we go. I mean, the videos are really large, and this is another video we have recorded, so to facilitate an understanding of how this works. So this is the top platform of you see like all the doors opens when the animal was first entered the central platform. Okay, the animal's about to enter the central platform and allowing access to all the different arms. And here we have the visual cues as well. And we are waiting a bit more, but yeah, sometimes the animal take their time. Okay, finally the animal's here. Yep. And then enters runs really fast. Enters, and this is a small space where we have the sugar pellets. The animal has a pellet and then returns to the central platform again. This is like a close-up of the pellet and the animal. Okay, there's no more to get, and then the animal returns. Okay, so I think this is these are helping us to understand how this thing mostly works. And of course we have some mechanical errors from time to time, but we have really like done a lot of trials to make sure it's within a controllable amount and it's not harming the animals. So this is a basis we want to secure before we start all the experiments. And then for the data analysis, of course, for those who have done experiments using automated maze or non-automated maze, we know these are things we want to analyze to explore the behavior on a activity level basis as well as a spatial learning basis. And since we are doing 2 paradigms only for the first half, or I think like 70% of the parameters we are analyzing for the first paradigm, and we chart on a daily basis how many visits are the animals making to the automated radial arm maze and how many arms are the animals entering per session. So these are the general activity level indicators. In addition to that, we want to quantify the effect of spatial learning. The first one is really important in the automated radial arm maze setting because we want to understand if the animals are really efficient, quantified by the amount of time the animal has to use to finish entering all the arms to get the rewards. In the second parameter being the number of beta arms entered. For example, for animals that have learned the setting and the spatial layout of the radial arm maze really well, they know like, okay, there are 8 rewards they can obtain, and for animals that did not learn that sufficiently, they're only entering a certain amount of the arms. And in addition to that, the most important parameter, I think, is the working memory error. It's indicating the entries again to visited and the baited arms in the working memory paradigm. And by the end, we are calculating the ratio of the percentages of the baited arms enters versus the total amount of arm entries. For example, if you're only entering 8 baited arms out of a total entries of 8 means you're making a really good, effective visits to these arms to collect the rewards. And on the other hand, if you're making a total entries of 100 and to only get, for example, 8 rewards, that's not so efficient according to us. And in the combined working reference memory paradigm, we also have 2 additional parameters we analyze. The first one is the number of entries to unbaited arms. So the unbaited arms, they are not returning any sort of sugar pellets. There's no reward to collect. So this is commonly referred as a reference memory error. In addition to that, we also calculate the number of re-entries to, for example, the baited arms, and I have a typo here. So the baited arms, and again, this is a bit of an invalid attempt because the rewards have been collected in these arms, although they were baited initially. So these are the parameters we are using for both of the studies. I think this also help us, making a standard for our analysis, so it's easier for us to compare even across the studies. Okay, so let's move on to the first study finally. And I think I spent a lot of time explaining how this whole thing works and how we are analyzing the data. So do we have any questions so far? If in case of no questions, I can just move forward. So for animals we are using for the first experiment is a total of 10 female mice and control mice. We're not doing any manipulations or any injections for this trial. We just want to make sure this is a proof of concept and the whole thing works well at a mechanical level, and we can definitely use it for further testing. This is a baseline, really the baseline study. So these animals are somehow old, and I think this is also like due to that we plan to use them for a different experiment at the beginning, and by the end they were just waiting and getting older. But this is also, on the other hand, helping us to know like if animals that are considered somehow older are good enough to use such apparatus to learn spatial navigation. And of course, we have RFID-based classification of animals, so we need this RFID transponder. So we do this implant at least 2 weeks before the experiments to make sure the animal have enough time to recover. And, sometimes if you have any experience doing the transponder implant, like if for the first couple of days, it can really easily slide out. So we also give the animals enough time for the implant to stay in their body without sliding out or falling out during the experiments. So we make sure that the animals are fully recovered and the IDs are working before our experiments start. And then regarding the experimental setup, one of the important thing is that our radial arm maze is controlled via a computer that's set outside the experiment room. So we don't need to enter them to really explore the different status of different mechanical parts of this radial arm maze. So this is one benefit of having such a thing. So during the experiment, there is no need for us to enter the room except for regular cleaning, because that's necessary because we have a group of 10 animals. So we definitely have to clean the radial arm maze from time to time. But other than that, we don't really have to enter the room, and there's no interruption or interference introduced by human experimenters. And in addition to that, we're really having a software to allow the regular monitoring of the radial arm maze. So we have animal technician or experimenters entering this corridor and staying out of experiment room again to make sure, okay, the radial arm maze is working and at all levels and all the mechanical parts, for example, the doors and stuff are all fine. And also via this software, we can see if there are animals within the maze they're overstaying, they're welcome. So in that case, we have to rescue the animal, but it doesn't happen. I think it didn't happen. But again, this is allowing us to explore the radial arm maze status outside the experiment room. I think that's quite important. And in addition to that, as I have introduced, it's quite important to introduce the animals to such a climbing piece and also sugar pellets and the taste and stuff like that. So we do in-cage habituation. The benefit of it is that they're staying in their home cage. We're just changing the lid. And they know the smell, they know their home environment, so they feel safe. They're just being curious, oh, there's something new added to my home cage, what that is. So that's the thing we are doing. And in addition to that, we typically record the session duration, which is the time the animal takes to finish visiting all the arms to get the rewards. But in case the animal are really staying in there for too long, first the animal can get panic or can be like some mechanical errors. In addition to that, the amount of time the animals for example, we have 10 animals in this case, as many animals. Actually, we did this in 2 batches. So we did it, I think, 5 to 5 or something. But if one animal stay in the maze for too long, we're really biasing this training received by individual animals. So we are keeping the max duration per session, like visit to the radial maze to 10 minutes. And after that, the animals actually, after this 10 minutes have elapsed, the animals forced out because we are closing the mechanical doors and the animal has only one way, if that is going back to the home cage. So for the first study, we do a habituation in the cage for 3 days, followed by the working memory paradigm, and then the combined working reference memory paradigm for 7 days and 5 days respectively. So for all the details regarding how this is done, you can see this in the papers referred at the bottom, and we have also attached the link to the videos we have had for the setup, and you can take a look if you want to, or you can talk to me after, if you cannot find it. So, and we also welcome any questions you may have. And this is how we have seen the experiments regarding the working memory over time and the combined working and reference memory over time. So if we took more indicators than this, but for others that we are not showing here, we don't see any significant difference across time. For example, I think for the daily entries and for some paradigms. So the first thing we can see that for all of these, we are showing generally a learning effect. So for example, for the animals, they can definitely learn about how we are getting more pellets within a shorter duration. So for example, the duration of session have actually decreased significantly over the 7 days of training. And while the working memory areas, the animal do not spend a lot of time re-entering the places they have visited in order to get the sugar pellets. So they learned that there's nothing waiting for them after their initial entry to these arms. So they're efficiently reducing the amount of time wasting and wandering around. And then, of course, after we have a reduced working memory errors, we have an increased baited arm out of the total entries because like the animals are not spending time blindly, entering and enter again the arms they have visited. And in the combined working and reference memory paradigm, we can see that the visits, because this is like a shorter paradigm, we can see like there's importantly, there's an increased amount of visits to the automated radial arm maze across the days. And in addition to that, we have significantly reduced amount of arm entries per session. So for this, we only baiting 4 out of 8 arms. So it's kind of important that the animals are not, again, like if they have learned the location of their respective rewarding arms, so they don't spend a lot of time just navigating around the arms and not giving any rewards. And the reference memory error is indicating that the reduced reference memory error is indicating that they're not visiting a lot of the arms that are not baited over the 5 days, while they have reduced re-entries to unbaited arm. So we think, given these results together, we have shown that the animals, even at 18 months of age, can achieve efficient spatial learning, although we have set the paradigms a bit short for the 2 paradigms, because we know like this is the first time we test such a cage, and we want to make sure the animals are fine and nothing wrong happens. And in addition to that, we occasionally have sometimes rarely we have mechanical errors, and we don't want to scare the animals and they're getting really old. So in addition to that, one thing that's really good about the automated maze is that it allows us to understand the activities of the animals across the 24 hours, 24/7 actually. But for the manual maze, we cannot really do that. I mean, some people may suggest that, okay, this is not the biggest concern we have, especially in spatial learning. But I think increasingly, as we are aware of the animals' welfare, as well as a correlation between the activity at a daily level and the performance in different learning and behavioral tasks, it's quite important for us to monitor the activities of the animals on a regular basis. So this is actually one benefit of the radial arm maze. It allows us to do that by having a automated tracking of the animal activity via the RFID. So as you can see, we definitely we are a typical animal lab, so we have this light and dark phases during the day, and so we call it inactive or active phase in correspondingly. And you see, like during the hour of the day, like where we have a more active phase, the animal have greater amount of visits to the radial arm maze, suggesting that we have increased level of activity. And I think in the future it's possible to correct this observation with the video recordings right above the the home cage. So in this case, we didn't do that because we have we don't have a transparent lid over the home cage, and we thought this is sufficient for this study. So, but still you can see like we have differential pattern of activity level across different phases. So in addition to that, because we have divided our, the animals, we also found some animals, they had activity at night, very curiously. So they are, sorry, not night, but during their inactive phase. So some animals occasionally visit the radial arm maze very actively during the active phase. So we also took a further step to understand how, the learning performance varied across the light versus dark phases. So we want to make sure like there's no big disagreement between these two. Otherwise, it's kind of curious. But as a result, we found for most of the parameters we're monitoring, we're analyzing, we find a general agreement between these activities during light and dark phases or learning during light and dark phases. But the only place we found a big difference is at the visits to automated arms. Of course, we have for the active phase, we have a smaller amount of visits to automated radial arm maze in both working memory as well as combined working reference memory paradigm. So this is like the only thing we found. Okay, there's a significant difference. Otherwise, there's a general agreement. We can just say this is caused by the pretty much by the activity level difference in the animals. And so I think that's pretty clear for the first part of our story. And as a summary, we can see like for the working memory paradigm, the animals we had learned to avoid visited arms. So they're making really efficient visits to the radial arm maze across time. And also they required less time to collect more rewards over time. That's really a happy finding. And working reference memory paradigm, we could say the animals had increased visits to the radial arm maze very curiously. And in addition, they have learned to avoid unbaited arms, which shows a decrease in reference memory error. And in addition, they did not really like avoid entering visited arms. So so we didn't really see a significant working memory difference in this paradigm. And what about if you're comparing our results to published studies using, it's really hard to do that because we don't find any fully automated maze published before in the radial arm maze testing using the same radial arm maze, of course. So we really struggled initially to find a study to compare with, but by the end we had this Miyakawa study published in 2001, where they use a semi-automated maze. So this is the closest thing we could find. And we found that the learning by the end is somehow comparable because the animal learned to visit all the arms and collect all the rewards and stuff. But it turns out the time the animals in our case took like when compared to their animals is actually shortened by more than 50%. So that's really encouraging, and eventually encouraged us to do the second study. So in the second study, we really we did our in-house comparison between the automated radial arm maze, or like instead of comparing to another study or something. I think this is the best way to take all the conditions under control and get the best of the experimental setups. So for this, we also explored how if we can use this apparatus to explore the effects of treatment. So in this case, we had a group that's LPS injected. So we know LPS can induce systematic inflammation, and we are really caring about, so this is like part of my PhD project where I care about long-term effects of systematic inflammation. So we also had a really long period before we started the experiments to see like the long-term effects of LPS on spatial cognition, of course. So it really aligns well with my PhD study. And for this, we had a greater amount of animals. We had 24 in total, and we randomized into two groups of 13 and 11 by treatment. And also we had younger animals that's around 2.5 to 3 month old. And for this study, we are having our own in-house classic or manual radial arm maze, where we have conducted, of course, a classic deprivation paradigm. We are also controlling that they're not reducing too much body weight because it's really confounding our studies. And for the manual radial arm maze, we do a 30-minute daily habituation. Of course, experiment has to do that, and we do this testing once per day. And overall, we have somehow a longer working reference memory paradigm for this study because we are sure that this is a safe apparatus, and we also want to really kind of understand how this learning plays out in the two groups over extended time period. And on the left, you can see there's a demonstration of how our classic radial arm maze look like. This is really classic. This is standard, and I think most people have seen that. So we have visual cues around the arms and like that. So these are done in the same room too, or like it's not exactly the same room because we have the two ongoing in parallel. So the experiment rooms we had in the corridor were identical. So we are kind of reducing also like the environmental effects on the study. And we are also contrasting these conditions. For example, like for the automated version of the study, we are again, the animal can enter this maze to up to 10 times per day. And in the meantime, we are, we are keeping all the arm entries monitored, and by the time the animal finished the whole experiment, we are encouraging the animal to go back with all the entry point to the arms blocked. And in the manual case, we have the animal collected and returned by the experimenter. Again, this is another level of human interference. Yeah, so we wanted to reduce it. And we did a cross design. So for example, as you can see on the figure on the right, we are doing these in parallel, and one group going through the automated radial arm maze, the other is going through the manual maze in parallel, and we just cross them when both groups finish. So we are having these studies going on at the same time, spanning across this amount of time. So for more details regarding how these experiments are done, you can refer to our paper in 2022. Yeah. So as a result, we can see that I mean, it's not too easy to see in this case, but I can try to explain again. So for the working memory paradigm, that's represented by for example, figure A and figure B. So this is the only place we found the difference between the groups. So for the fourth day of the working memory paradigm, we found there's a group difference indicating the treatment effects as only the last day of it. And also there's a difference by the correct entries to the radial arm maze, to the arms in the radial arm maze, which is, we have greater % of correct entries by the control group. So these both indicate that the LPS treatment has some effect on the spatial learning abilities in the animals, as they are demonstrating worse working memory, and as indicated by the working memory error and the correct entries ratio. But for the combined working reference memory paradigm, as you see in the last 4 subplots, there's no significant difference across the groups in the automated radial arm maze. And then again, for the manual version of the maze, what we see is kind of interesting, because if you recall from the previous slide, we have seen some differences in working memory paradigm for the automated radial arm maze, but for the manual version, we didn't see any differences during that phase. But all the differences we have seen are from the working reference memory paradigm indicated in the last 4 subplots. So again, for the working memory errors during the combined paradigm, we have a difference between the groups on the last day, and the data are log transformed for the analysis for these p-values. And for the reference memory errors, we had a difference between the groups on the first day of the experiments, as well as for the correct ratio, ratio of the correct entries. So this is demonstrating the opposite effect. So for example, for the automated radial arm maze, we have only seen the differences between groups for the first paradigm, but for the manual version of it, we have seen for the second paradigm, which is quite interesting. And again, we monitor the animal activities, but I think this is somehow is not as prominent as that one. It's somehow a bit mixed up, like the trend is not as obvious as that one for both groups, but still, like during the more active phase of the hour of the day, we can see somehow a certain level of increased activities in both groups, meaning that they have regained active day-night pattern, which is not the case right after the LPS injection, as shown in the other study of my PhD thesis. And so if you're comparing, by the end, we're also comparing how like the general aspects of these two versions of radial arm maze are. For example, if you're taking account the animal stress level, we can see that the automated version of is actually lower because we don't really need a lot of human interference during habituation and testing and training phases. Well, in the manual version of the radial maze, we need to habituate the animal on a daily basis prior to experiments, and during experiments we have to make sure we are putting them in the radial maze and collecting them by the end, which is somehow really panic inducing because you're kind of chasing the animal and grab by the tail. So it's really like a greater level of animal stress. And we didn't need any food restriction during automated radial arm maze for the animals, and their overall physiological state did not change at all. And for the manual version, we definitely, in our case, we also did the food deprivation, which is necessary to motivate the animal to achieve enough motivation to navigate within the maze. And again, in corresponding to the first point, the third, the animal experimental interaction also was really low in the automated version of the radial maze. Well, because we have a lot of contacting points between the two parties, we have a higher level of animal experimental interaction in the classic or manual version of the radial maze. And the daily effort we are spending in the automated version, I mean, of course, there's daily effort because we have to make sure the setup is all fine, and even considered, using remote monitoring, from home to monitor the animals. But it turns out they were safe overnight, so we dropped this idea. But the daily effort, we have to spend a couple minutes per day to inspect the maze while the software's interface. Well, in the manual radial arm, we have to have the experimenter, spending hours to test the animals, especially when you're having a large amount of animals for testing. So in our case, it's around 24, but in some other cases, we want to have a bigger N, or if you are conducting bigger multicentric studies, it's actually, increasing constantly exponentially, but significantly compared to the automated version of the radial arm maze. So then again, it's possible to assess the day-night rhythm and level of activities in the automated radial maze, which is getting increasingly important in animal welfare studies, also baseline experimental studies. However, it's kind of impossible to do this in a manual radial maze. You have to complement that with for example, about the head video recordings or something like, or co-registration of ID transponders and, a sensor table plus something from above to record the animal's activities. And what else we have to mention about the automated version of the radial arm is that the data can be really high, because although we try to motivate the animal as much as possible for them to enter the radial arm maze, it's not like always working. And for example, for the second study, we actually excluded a lot of animals in the first part. It's because they did not visit the automated radial arm maze for sufficient amount of time per day, so the learning was not efficient. So it really, this is really a variable we have to consider when we are trying to incorporate automated radial arm maze. Of course, I'm open to suggestions. If you have a better suggestion how to motivate them better, yeah, of course. Or we can do a hybrid approach where we are motivating them through a milder food restriction, but I don't think that's quite desirable. And in the meantime, the manual radial arm maze is not really having a data exclusion as a big component of it, because we are kind of doing this manually on a daily basis, and it's really, the margin of error is not somehow comparable to the automated version. And by the end, if you wanted to set up an automated radial arm maze, it's really like a dedicated device, and you have to really make sure that the animal technicians are okay to inject these transponders, and you have to have someone inspecting the software interface. You have to coordinate with people installing it to make sure there's no mechanical error. I think I spent right before my PhD is already being installed, and we spent a month really monitoring and inspecting. Okay, everything's fine. We even took the time to do the rounds to check the sugar pellet dispensers to make sure it's all dispensing 1 pallet per time exactly. So it's actually startup, mechanical and personnel costs for this is definitely higher, but for the classic manual version, it's just standard. So it's whatever you spend on a manual radial arm maze. So I think it's all, they're having their respective merits in the ways you see that I'm not just blindly advertising the automated version, but we have to consider more comprehensively what you're getting out of what you're paying for. So as a summary of the second step, we can see that for the working memory paradigm, we can see there's only worse performance by the LPS injected animals in the automated radial maze, and the story is the opposite for the working reference memory paradigm. In addition to that, the exclusion of data is really high for the first paradigm because we had animals that are I think lower motivation in general to enter the radial maze or feel there's no need to. So for 24 animals, there were 8 we excluded from both groups. And in addition, in both paradigms, we had software errors, and there's leading to a certain percentage of exclusion. I think for the first study is around under 5%. So I personally conducted the first study, and the second study were conducted by the first author of this paper. So we also compared notes, and somehow the software error is still a big thing, and you want control for if you're trying to automate the whole system. And by the end, we could see that when contrasting with previous studies on how LPS is affecting spatial learning performance in radial arm maze testing, we had actually mixed results. We have these 2 papers reporting different things. But for our study, we are using a lower dosage of LPS, which is around 1.5 milligram per kilogram. And this is leading to somehow subtle treatment effects, but we could still see some differences. And it's also corresponding to another spatial learning study I've done during my PhD. So there's definitely some level of effect, but depends on the storage, on the dosage, and the setup cannot be so significant in all time points. And so overall, like through these 2 studies, we can see that we can fully automate the radial arm maze, and during this requiring us to potentially set up a habituation cage and do the ID sorting for the animals' entries to the radial arm maze, and also have automated version of activity tracking, so we know like which animal has entered which arm at what time points and how many rewards the animal has collected, etc. And in addition to that, we have, I think for most of it, especially for the first study, we have voluntary entries by the animals. And I think it ranges from 2 entries or 2 or 3 entries per day to up to 10 entries. If you're not limiting it, I think for the trial, like there's one trial period, we didn't limit this at all. So animals can enter up to 20 times, I think, really depending on the group size as well. And we have seen animals, they're trying to, compete to enter this. And sometimes we have 2 animals in ID sorter. That's quite interesting. And also, more importantly, we have seen effective learning in the 2 paradigms using the radial arm maze, although we did not really fully deprive them to achieve this general motivation. And when we are comparing this with the manual version of the radial arm maze, we can see that we didn't really need to restrict any food or water intake, and also we minimize the handling, in this testing, which is a good thing. And in addition to that, one disadvantage of the radial arm maze that's automated is that the data accumulation can be really high due to insufficient motivation or due to software errors. And I think for my experiments, I also had a bit hardware errors as well. And when you're comparing the 2, especially from the second study, you can see they have their respective mirrors and they can detect the learning differences at different stages. So that's quite important for us to remember. And by the end, we also validated the usability of such a device for different age groups, and they're ranging from 3-month-old to 18-month-old, like young animals to old animals, and they were all learning really fine this setup. And we were able to review the treatment effects of LPS also very subtle. So I think that's pretty much all I want to talk about today. And thanks a lot for your attention and interest. And I really want to know if you have any questions or concerns to share with us. Thanks.